So today I will be presenting one of the projects I've been working on during my postdoc in Jeremy Purvis's lab at UNC Chapel Hill, where we're combining single-cell imaging techniques and computational approaches to try to generate a new updated map of the human cell cycle. So we all know that when I say cell cycle, I'm talking about the ordered sequence of events that a cell goes through to first replicate its DNA and then split it between two genetically identical daughter cells through cell division. But what does the internet think that the cell cycle is? As you can see, the cell cycle is almost universally represented by this circular structure that breaks up this process into four distinct phases. In G1, when the cells grow and prepare to replicate their DNA, S phase, where the DNA replication actually happens, a subsequent G2 phase, where the cell checks for errors made during the replication process, and finally, mitosis, when the cell actually divides in two. And sometimes the structure will also include this little loop here, usually somewhere in G1 into this other phase called G0, where the cell just rests, apparently. Now, I'm joking a bit here, but the truth is that this structural representation of the cell cycle has been a very powerful tool over the years, allowing us to map the molecular events that govern passage of a typical cell through DNA replication and cell division. Now, our current understanding of the cell cycle has been assembled piecemeal over several decades of careful biochemical and genetic experiments. And often these experiments involve chemically synchronizing cells in specific phases of the cell cycle, and then taking bulk measurements from the entire cell population. Now, while these approaches were extremely productive for many decades and continue to be very useful today, like in many other fields of cell biology, the development of single cell techniques over the last decade or so is revealing novel aspects of cell cycle biology that were just not accessible using these bulk population measurements. And most of this single cell work in the cell cycle field has come so far from the use of fluorescent biosensors and single cell time-lapse imaging, starting with the development of the original Fuji sensor a little over a decade ago, which allowed individual cells to be tracked as they progress through each phase of the cell cycle. Now, using Fuji or some of the newer iterations of it, such as the PIP-Fuji sensor that we designed in collaboration with Gene Cook's lab here at UNC, we see a great deal of variability in how long it takes individual cells to pass through each phase of the cell cycle. Now, the field has also developed a number of biosensors that monitor the expression and or the activity of key cell cycle regulators, which in combination with these cell cycle sensors are now being used to try to understand exactly how this heterogeneity in cell cycle progression is being generated. Now, at the heart of all of these studies is really this question of what is the topology of the cell cycle? Is this model really true that cells proceed through a single fixed order of molecular events? Or maybe cells can take different mechanistic routes through the cell cycle. I mean, does the choice of route determine how rapidly an individual cell progresses through replication and eventual division? Now, a road trip is an easy analogy here. Let's say this road trip I usually take around this time of the year from North Carolina to go back and visit my family in Canada. I can enter my destination into Google Maps. Google will offer me several routes to choose from. Each of these routes has a different intrinsic length. There are also different points of divergence where I can make a decision between one route or another. And there are even possible roadblocks that might influence the path that I take. Now, ultimately, the time it takes me to get home will be determined not only by how fast I drive, but also which route I've chosen and whether I ran into any problems along the way. In the last few years, we've realized that cells encounter similar decisions on their road trip to mitosis. We know that if cells encounter stress during G1, they may revert back to an earlier state of the cell cycle while they cope with it, and then restart their journey once again. If cells are treated with a CDK4/6 inhibitor, which blocks the canonical path through G1, they can actually take a detour through a different mechanistic route that takes a little bit longer, but has no effect on the passage of the cell through the rest of the cell cycle. And perhaps the most established example of cells taking different routes through the cell cycle is the fate decision of daughter cells immediately following mitosis. A very nice series of papers using a suite of cell cycle biosensors has demonstrated that following mitosis, daughter cells can either immediately re-enter the cell cycle, they can temporarily exit the cell cycle into a state of arrest, and then come back after some variable amount of time, or they can permanently exit the cell cycle. Now, these studies have demonstrated that some of the observed variability in cell cycle duration is determined by which of these fates a given cell takes at the very beginning of its cell cycle. And although cell cycle biologists are starting to appreciate more and more the role of this G0 state in cell cycle fate decisions, we still don't understand some of the very most fundamental aspects of this cell cycle arrest state. It is still unclear, for example, whether G0 is a single arrested state, or as it's depicted on many of the models that Google showed us earlier, or is it rather a collection of cell states? And although we have some understanding of the mechanisms that induce cell cycle arrest, it's still not entirely clear when this decision is made, or more precisely, when do cells diverge from the proliferative cell cycle into a state of arrest? And finally, what happens to cells after they exit the cell cycle? Do they remain in a static G0 state awaiting cell cycle reentry? Do they enter progressively increasing depths of arrest? Will this arrested state eventually turn into an irreversible arrest or senescence? In essence, we know very little about the topology of cell cycle arrest. And so while the use of fluorescent biosensors and time-lapse imaging have been very powerful in elucidating specific aspects of cell cycle regulation, we wondered if we could design an experiment that would provide a sort of a bird's eye view of the entire cell cycle to try to provide a more comprehensive map of the paths that cells can take, including the paths that lead to cell cycle arrest. Now, in order to obtain the most complete roadmap of the cell cycle, we need to design an experiment that satisfies a few criteria. First, we need to design an experiment that can sample cells in all possible states of the cell cycle, even rare states. Secondly, we need to find an assay system that allows us to make measurements of the cell cycle state in each of these individual cells. And third, we need to find a way to order the cells by their cell cycle state signatures in a way that provides a visual representation of the cell cycle and the molecular trajectories that go through it. And we reason that we should be able to capture the full diversity of cell cycle states if we simply sample a large enough population of asynchronously cycling cells at any given time point. So we started with a population of around 10,000 asynchronous human retinal epithelial cells, or RPE cells, which is a non-transformed human cell line that was immortalized with telomerase and contains a sort of normal repertoire of cell cycle regulators. We then performed time-lapse imaging of this population using a single fluorophore reporter of cell cycle progression, so that we had a chronological measure of how long each of these cells has spent in their current cell cycle, as well as the knowledge of the cell cycle phase that each cell was in at the end of the movie. Now, we now need to make a measurement of each cell's cell cycle state. Usually these higher order representations of cellular state are made these days by single cell transcriptomic measurements. However, many of the mechanisms known to govern cell cycle progression are driven primarily by the degradation, post-translational modification, the subcellular localization of various proteins, which cannot be captured very well by transcriptomics. So after time-lapse imaging, we fixed the cells and performed iterative indirect immunofluorescence imaging, or 4I, on the cells. This technique, which was developed by Lucas Pelkman's lab in Zurich, is one of several being used right now to perform multiple rounds of immunofluorescence on the same sample, or on the same population of cells. In 4I, this is accomplished by stripping off the antibodies after each round of imaging, and then reprobing with a new antibody set. In this experiment, we used 4I to measure the expression and or the activity of 47 different cell cycle and signaling effectors in this asynchronous population of cells after time-lapse imaging. Now, one key advantage of imaging-based proteomics is that we can derive additional features, in this case around 3,000 unique features for each cell from the images we obtain. Now, these derivative features include things like the subcellular distribution of proteins, the size and shape properties of the cells themselves, as well as features of the cell microenvironment, things like local cell density. At the end of this experiment, we ended up with these high dimensional signatures representative of each cell's cell cycle state for almost 9,000 individual cells. Next, we need to find a way to visualize these high dimensional data in a lower dimensional space to try to make sense of them. Now, our assumption here, of course, is that cells at similar positions of the cell cycle will have similar cell cycle signatures and be located close to one another in this high dimensional space. And our job is to find these trajectories through the high dimensional space, this manifold, and project it down into lower dimensional space. Now, we've played with a number of the so-called manifold learning techniques, and we settled on an approach called FATE for this experiment due to its strengths at preserving the continuous branch paths that we expect to find in the cell cycle. Now, using FATE, we obtained this continuous fork-like structure from this experiment. We can then overlay the data that we obtained from our time-lapse imaging directly onto this structure, as shown here for cell cycle age. Now, as you can see, the youngest cells from our time-lapse imaging mapped directly on the central arm of the structure, and cells age as they progress up both the right and left arms of the structure. If we overlay cell cycle phase information on the landscape, we see that G0 and G1 cells occupy the left and central arms, while cells progressively passage through S and G2 phase along the right arm of the structure. Now, one important thing to reiterate at this point is the structure was derived from the 4i cell cycle signatures alone, and that none of this information that was derived from time-lapse imaging was used by the model to create this landscape. So the fact that cell cycle phase and annotations and age annotations seem to line up in a very clear way that makes sense, it's actually just an indicator of how well this completely data-driven approach performed at finding real cell cycle trajectories. So we have G0, G1, S, and G2 phase cells mapped on the structure. But where are the mitotic cells? Now, unfortunately, M phase cells are underrepresented on our structure, and this occurs for a couple of reasons. First, since M phase is relatively short compared to the other phases, at any given fixed time point, there are simply not that many cells in the population that are actively undergoing mitosis. But the second reason is that the M phase cells are disproportionately lost during 4i relative to the other phases. And we think this is likely due to the reduced surface adherence of cells during mitosis. However, the single M phase cell that was annotated and persisted throughout 4i resides right here on this central arm of the structure. So to help figure out more precisely where mitosis is located on the cell cycle landscape, we designed a tool that allows in situ visual inspection directly on the structure itself. And sure enough, we found that cells along this central arm are just finishing cytokinesis and entering into the earliest stages of G1, consistent with this region also being the region where we find the youngest cells in our population. We then wondered if this projection from the G2 area of the right arm here might represent cells in earlier stages of mitosis. Indeed, we find that cells along this trajectory show increased nuclear cyclin B, an increase in DNA condensation, and eventual nuclear envelope breakdown. So although we have an underrepresentation of mitotic cells on this landscape, we do successfully capture cells in early prophase and in late telophase. And we're quite confident that our missing metaphase and anaphase cells likely reside somewhere along a trajectory here between the rightmost arm and the central arm of our structure. Now, you may have noticed that earlier, when we presented this data, we only have annotations from time-lapse imaging for a subset of the total cells that we performed 4i on. Now, this is because we need to limit our time-lapse imaging to only a subset of the total well area that we image for 4i in order to preserve decent time resolution for the movie. If we had imaged all of the cells that we eventually performed 4i on, our frame rate would have been lower than we'd like to accurately delineate the transition between phases. To account for this, we decided to train a random forest classifier using cells for which we had both time-lapse imaging annotations as well as their 4i signatures, and use it to predict the cell cycle phase of untracked cells. Now, unfortunately, we can't distinguish G0 from G1 cells directly from our time-lapse data using our PCNA sensor. But we can use the information obtained during 4i to delineate between these two phases. The phosphorylation of RB is often used as the molecular correlate of cell cycle commitment, and it distinguishes G0 from G1 cells. We see a very clear delineation along the central arm right here between cells with phosphorylated RB and unphosphorylated RB. Now, this dividing line provides a very clear visualization of what is referred to as the cell cycle restriction point on this landscape, which separates actively cycling cells from arrested cells. We also use a random forest model to predict cell cycle age. And we see here clearly that the cell cycle age increases as cells progress through G1, S, and G2, but also along this arm of G0 arrested cells. Now, altogether, we have a pretty clear idea of how to interpret our cell cycle landscape, which seems to be made up of two trajectories. We have first a cyclical proliferative trajectory, starting here along the central arm, and progressing along the right arm through G1, S, and G2 before looping back to the central arm during mitosis. And along this leftmost arm, we have a rest trajectory, with cells spending longer and longer amount of time in the arrest state as they progress further and further up this arm. So I'd like to also point out that we obtained this same general structure over a wide range of hyperparameters that could be tweaked in FATE, which is really nice to see. And we also find a remarkably similar topology when we perform the same experiment in a completely different cell line. These HP&E cells here, the human pancreatic epithelial cells. So these are all very promising signs. So next we asked if we could capture known cell cycle biology on this landscape. We decided to take a look at whether we could visualize specifically the molecular mechanisms that control the G1S transition. Now, the core unit regulating this transition is this inhibition of E2F transcription factors by the RB protein. Now, relief of this inhibition and commitment to DNA replication is driven by the phosphorylation and inhibition of RB by cyclin CDK complexes. Now, in early G1, RB phosphorylation is performed by cyclin D CDK4/6 complexes. And we observe that cells begin their cell cycle with high cyclin D1, and it decreases as cells transition into S phase. This cyclin D-driven inhibition of RB then initiates E2F-mediated transcription, which starts to increase in G1 and remains elevated throughout S phase. One important E2F target gene is cyclin E, which begins to rise in late G1 as E2F-mediated transcription ramps up. And by forming a complex with CDK2, cyclin E helps compensate and maintains RB phosphorylation as cyclin D levels begin to drop. Now, another important event in the G1S transition is the inactivation of APC complexes, which act to degrade and prevent the accumulation of S-phase proteins. This increase in cyclin E CDK2 activity in late G1 stimulates the destruction of the APC subunit CDH1, which then allows S-phase initiation. The inactivation of APC complexes also allows another important S-phase gene, cyclin A, to accumulate as S-phase begins. Cyclin A CDK2 complexes then take over the RB phosphorylation duties as cyclin E levels begin to drop over the course of S phase. Now, of course, we observe also the most important event of S phase, DNA replication. We see that DNA content roughly doubles over the course of S phase. Now, altogether, this demonstrates that the topology that we capture in our cell cycle landscape not only incorporates, but is in fact, shaped by the molecular mechanisms that govern progression through the cell cycle. Now, as I mentioned earlier, most of what we know about alternative cell cycle trajectories comes from work studying the fate of daughter cells following mitosis, with some cells immediately re-entering the cell cycle and others spending a brief stint in G0. And if we highlight the very youngest of our cells, we see a clear bifurcation of cells following mitosis either into G1 with high RB phosphorylation or into G0 with low RB phosphorylation. These cells that head down the arrest trajectory also show an increase in the expression of P21, which induces RB dephosphorylation through the inhibition of CDKs. If we plot out RB phosphorylation by cell cycle age, we see that all cells seem to end mitosis and begin their cell cycle with high RB phosphorylation, and then diverge into two populations, one that maintains high RB phosphorylation and enters into G1, and another where RB phosphorylation dramatically drops and cells enter this state of arrest of low RB phosphorylation. Note that these cells are also colored by P21 expression. And so we see that this drop in RB phosphorylation entry into G0 corresponds very well to an increase in P21 expression in these cells. So while this seems to be one way that cells can enter the can exit the proliferative cell cycle into a state of arrest, we also see some indication that cells that are expressing high levels of P21 in G2 phase might actually exit the cell cycle here and enter into a state of arrest further down this arrest arm here. Now, unfortunately, since this experiment was performed on cells growing under ideal culture conditions, it wasn't necessarily designed to get good coverage of the arrest states. So it's hard to make any real solid conclusions about these arrest trajectories from this data alone. So to better explore the topology of cell cycle arrest, we performed another 4i experiment after subjecting cells to different types of cellular stresses that are known to induce cell cycle arrest. So first we subjected cells to hypomitogenic stress by growing them in serum-free media up to 7 days. I would like to point out at this point that once again, we obtained a similar fork-like structure in control cells consisting of a cyclical proliferative trajectory here and an arrest trajectory extending from this middle arm. Now, under serum-starved conditions, we see the appearance of this new population of cells with low RB phosphorylation and 2C DNA content, indicating a G1-like arrest state. As you can see, this hypogenic arrest state is distinct from the spontaneous arrest state observed in control cells here. Now, furthermore, this exit from cell cycle following serum starvation is driven by a decrease in cyclin D expression in G2 relative to control cells, which is consistent with the fact that one of the main ways that mitogenic signaling drives cell cycle progression is by inducing cyclin D expression. Now, according to our map, serum-starved cells diverge from control cells in G2 and undergo mitosis directly into this arrested state. And unlike the spontaneously arrested cells that we observe in control cells, this hypomitogenic arrest state does not appear to be driven by an increase in P21, but instead we see that progression further into this arrest state is driven by an increase in the related CDK inhibitor P27. So in summary, hypomitogenic arrest seems to be a single graded arrest state that is driven by insufficient cyclin D expression during G2 and is then stabilized by an increase in P27. Next, we assess the effect of replicative stress on cell cycle progression. Here we see that treated cells, here we treated cells for up to 4 days with etoposide, a chemical which prevents the religation of DNA during replication to induce DNA damage specifically in S phase, thus mimicking the effect of endogenous natural replication stress. As you can see, the arrest architecture after replicative stress is far more complex than the single arrest state that we observed after serum starvation. We observe this trajectory that loops between G2 and the arrest arm control cells. And with increasing days of etoposide treatment, we see that cells gradually disappear from this inner loop of the arrest states here and start to accumulate on this outer loop. Now, while the majority of these cells, and along all the loops, exhibit low phospho-RB, indicating cell cycle arrest, we see that these upper cells actually have a 4C DNA content, indicating a G2-like arrest state, while these lower cells have 2C DNA content, consistent with the G1-like arrest. Furthermore, we see no evidence of mitosis, that mitosis is actually occurring between these two populations, indicating that in response to replicative stress in S phase, cells can exit the cell cycle along two distinct arrest trajectories. Either here in G2 immediately following the acquisition of DNA damage, or here after mitosis and in the subsequent G1 phase of daughter cells. Now, consistent with this, it's a mechanism of action, we observed that following etoposide treatment, both of these states of arrest are driven by increases in DNA damage signaling, as shown here by increases in gamma H2AX, which is a direct readout of the magnitude of DNA damage, the activation of CHEK1, which is a component of the DNA damage response machinery, and increased expression of P53 and P21 as transcriptional target, which are, P21 is the protein that sort of couples the DNA damage response to cell cycle arrest. We also see a lot of overlap between this 2C arrested state and the spontaneously arrested state that we observe in control cells, which, remember, is also driven by an increase in P21, suggesting that this spontaneous arrest state is likely related to the acquisition of mild natural replication stress in control cells. Now, we also see that as cells progress further along this arrest trajectory out of G2, they abruptly shut down the expression of S and G2 cyclins, cyclin A and cyclin B, and they start expressing the G1 cyclins, cyclin D and cyclin E. Now, this normally occurs as cells complete mitosis and re-enter a new cell cycle. However, these cells don't even attempt mitosis and instead transition into a G1-like state without even dividing their DNA content. And this is a phenomenon known as mitotic skipping. We then wondered if cells could re-enter the cell cycle from this state, given that along this trajectory here, we see an increase in cyclin D first, followed by an increase in cyclin E, which is the same sequence that we see in a normal G1 phase. Now, consistent with this fact is that we observed the appearance of polyploid 8C cells only after several days of etoposide treatment, which we believe are cells that re-enter the cell cycle after skipping mitosis once. They then succumb to etoposide-induced DNA damage once again while trying to replicate their already doubled genome, and then exited the cell cycle along this G2 trajectory here. So in response to replicative stress, cells can exit the cell cycle along two distinct trajectories, either in response to checkpoint activation in G2 or after mitosis in the daughter cell G1 phase. Furthermore, we see that cells that exit from G2 can undergo mitotic skipping and re-enter the cell cycle, a process that can induce polyploidization. Now, finally, we have oxidative stress. Now, the dominant cell cycle effect that we observe in response to oxidative stress seems to be primarily related to its ability to induce DNA damage. Now, similar to etoposide treatment, we observe cells exiting G2 with high levels of DNA damage and entering a state of arrest with 4C DNA content. We also observe some cells entering a 2C arrest state directly from G1, just like we did with etoposide treatment. So now you can believe me that these are the same as the arrest states that we see following etoposide treatment, but it's much easier to see it on a map that includes all of the stress-induced arrest trajectories at the same time. Now, presented as a single landscape, you can clearly see that cell cycle arrest is a complex architecture of molecular trajectories. We see that replicative and oxidative stress induce similar architectures, with cells exiting the cell cycle in response to the G2 checkpoint and undergoing mitotic skipping, or after completing mitosis in the daughter G1. This spontaneous arrest state that we observe in control cells also appears to lie along this trajectory, consistent with a low level of natural replicative stress. And we see that serum starvation induces cell cycle exit along a completely different trajectory into a single graded arrest state. We also outlined the mechanistic trajectory through which sustained DNA damage can induce polyploidy following mitotic skipping. Now, altogether, through a completely data-driven approach, we were able to generate an updated map of the human cell cycle that both captures known cell cycle biology and provides a more comprehensive description of the molecular architecture of cell cycle arrest, including multiple points of divergence from the proliferative cell cycle into distinct states of arrest in response to different cell cycle stresses. And with that, I'd like to thank Jeremy and the rest of our awesome lab. In particular, I'd like to thank Kasia, Colin, and Tarek, who were instrumental in helping progress the computational elements of this project. And I'd also like to thank Holly, Catherine, and Sophie, who really kicked butt at the bench helping to generate the crazy amounts of data that went into this project. I'd also like to thank Jean Cook and her lab for all of the help and the reagents they provided along the way to push this project forward. And I'd be happy to answer any questions that you might have at this point. Thank you very much.